Hi guys, so Tom Bannon's my name. So I'm a process SME with PM Group. So PM Group is we're nearly a 3,000 strong architecture and engineering firm, and we offer services right across biopharmaceutical manufacturing and increasingly more cell and gene therapy projects in, let's say, in the last 5 or 6 years. So my personal background is I'm a process engineer by training, and masters in biopharmaceutical manufacturing, and I've been with PM Group for the last 10 years. And I suppose my areas are generally anything biologics manufacturing, and kind of more focus on allogeneic and autologous cell therapy facilities. Do you want to introduce yourself, Arantxa?
Yeah, okay. Hello, my name is Arantxa Lera. I'm an architect by background. I have a master's in architecture, but also a master's in urban design. And I've been with PM Group for 8 years now. Most of that time, I've been the head of architecture in the UK, and I recently moved to Ireland in an SME role, where I've been supporting projects globally. And just as projects went little by little, I started working more and more in cell and gene therapy projects, and that's what the presentation today will focus on. So over to you, Tom.
Okay, so give us the next slide, Arantxa.
Yeah.
So just our talking points for today. So we're just going to set sort of some context initially about the future of cell therapies, and I suppose we're going to talk about how that today it's a lot of autologous cell therapy, but in the future it's going to have a lot more allogeneic cell therapy. So I will describe those two processes and what they mean in the coming slides. So then once I've done that, I'm going to hand over to Arantxa, who's going to talk a little bit about how we design these facilities, and then how we design the facilities today that they can be of use for allogeneic facilities into the future. But Arantxa is going to pick the back half of the presentation. Okay, Arantxa, I'll take the next slide.
Yeah.
So at the moment, I'm guessing there's only about 7 or 8 cell therapies approved in Europe at the moment. There's only, sorry, actually no, there's 9. So there's only one allogeneic cell therapy, and that is a product called Allogeneic by Takeda. While the majority of the other cell therapies, now this is for Europe only, are the CAR T therapies, which are getting a lot of publicity at the moment for, I suppose, revolutionizing how we treat blood cancers. So that's the current state of play of what's approved today in Europe. There's only one allogeneic cell therapy and approximately 8. I think 7 of them are CAR Ts, and then there's another one treating something else. So I suppose the big difference, just so people get the context, so autologous cell therapies, where your own cells are taken, are modified, and then your cells returned back to you, while allogeneic is where the cells you're receiving as a patient are actually cells that belong from somebody else. But I'll get into that in a moment. Give me one more click, Arantxa. So if I actually take just, this is, I want you to take this as very, very high level, because a lot of the definitions of what the US, for example, call a cell therapy versus what Europe calls a cell therapy is quite different. So just take this slide purely as a theme. But in Europe at the moment, roughly 1 out of 10 approved cell therapies is allogeneic. But if I look at the UK, and the reason I've picked the UK is that Cell and Gene Therapy Catapult publish a very comprehensive sort of clinical trials database of what is happening in their country in terms of clinical trials. So if I start working back from approved and I look at phase 3, so currently in phase 3 in the UK, there's roughly 30% of what's in that stage of clinical trials is allogeneic, with 70% being autologous. And then if you work further back again into the phase 1, the very early stages, you're seeing a lot more allogeneic cell therapies coming down the future. So roughly 60-40. Now, this is very, very high level, and I'll explain a little bit later on, because what cell therapies are treating is a huge, wide base. So it's not that all CAR Ts, for example, are in allogeneic at phase 1 at the moment, but I'll explain that in a moment. Okay, so give us another slide, Arantxa. Okay, so what's the difference? What are the two differences between the two types of processes? So if I take the little graph on the right, the very start of the manufacturing process, the top of the slide, is the patient. The patient is in hospital, they're quite sick with some disease, and we need to get a donation from that patient of cells that's going to leave that patient, get brought to a manufacturing facility, have a modification done to it, and then returned back to that patient. So give us one click there, Roger. So the key thing with an autologous cell therapy is that it starts with the patient and it ends with the patient. The patient must get their cells back. And the reason there for that is that there are certain therapies that what you need to avoid is graft versus host disease, where the patient will reject foreign cells that don't belong to them. Okay, so a lot of the CAR Ts today are autologous, so that CAR T blood cancer therapies that are many of them are approved in the US and Europe. They are autologous today. So what that means, if it's one patient, one dose, that means I don't scale up my batch. I don't make a batch and I grow more cells, and then that one batch can treat hundreds of patients. It's one patient, one dose. And that's what is the main logistical challenge of these types of therapies and their manufacturing. So because it's one patient, one dose, the batch sizes are, they often don't exceed 10 liters. That's the biggest scale you get to. So that the largest bioreactor we have seen in autologous cell therapies is a small wave bioreactor. So everything is benchtop scale. But some of the more recent facilities that we're designing, the working volume does not exceed 500 mil in any sort of bioreactor systems that are being used. So it's benchtop equipment, very small equipment. There's often a lot of very manual manipulations required on the cells. So this means we're working in biosafety cabinets and using aseptic technique quite a lot. Again, I've talked about the batch size. So this is the key point at the very end of the process, is that this is a drug product at the end of the day. This is going to get infused into a patient. So they have to formulate this. So what they add is DMSO, which is a cryopreservant, because when I freeze the cells and cryopreserve them to ship them back to the hospital, they need to be, a cryoprotectant needs to be added, and DMSO is traditionally what's used. A little point here is that DMSO, in the liquid phase, DMSO is toxic to the cells. So the second I add my DMSO, I very quickly have to finish the formulation. I need to fill into my final containers. I need to visually inspect the final containers, and then I need to load them into the controlled rate freezers. And then once that happens, they're frozen. And the key thing I want to highlight there is that once they're frozen, it's very difficult. In fact, you can't complete the visual inspection at that point. So in autologous cell therapy, usually that's okay. And the reason it's okay is that you roughly have about 2 hours from the moment you add your DMSO to do that formulation, to do that visual inspection and loading into your controlled rate freezers. And again, in our experience, while the patient's going to receive one dose, there's probably a second backup dose, and then there's probably maybe 10 or 15 vials that are taken away for QC and retains and legal retains. So the actual filling and formulation isn't that complex a task, and they can do it in the 2 hour time window. So just remember that the DMSO and the 2 hour time limit is key for the next couple of slides. I'll take the next slide, Arantxa. So what's different with allogeneic? So with an autologous cell therapy, as I said, it leaves the patient, you do your manufacturing process, and then you return it back to the patient. And that way the patient avoids any graft versus host reaction. So what does allogeneic cell therapy does? So often there is a further, one of two things can happen. One is the patient, after they receive the therapy, are put on medication to prevent rejection of the cells, exactly like a donor who receives a kidney transplant. So what this means is that you have to balance then the decision to say, is it more beneficial for a cheaper cell therapy where I can make multiple doses for multiple people, but then anti-rejection medication has to be given to the patient? Or what is now emerging more and more is that they further genetically modify the cell therapy so that it won't get rejected by the patient. So now you have the cell therapy having two modifications done to it, one to treat the disease and one to prevent it from being rejected, okay? And once that happens, that opens out that we can make a batch that will treat multiple patients. So an allogeneic cell therapy will start with, it'll either start with a donor who regularly donates, okay? So it might be one person who maybe once a month or once every 6 months is donating cells, or you may have some form of characterized or engineered cell bank, similar to traditional biopharmaceuticals. But what we're more seeing is what they call cell stock. So they have regular donors donating cells to be the start of the manufacturing process. And in this case, then they do have the opportunity to expand the cells, grow multiple cells, and formulate that, fill it in a traditional filler, and now they can make enough doses that maybe one batch can do, rather than one patient like Autologous, Allogeneic could do maybe hundreds or even thousands of patient doses. So this makes the cell therapy much more affordable and brings the cost, manufacturing costs right down. So as I said in Autologous scales out, Allogeneic scales up, okay? So we get more doses per batch. So we get larger bioreactor systems. So I'm saying 50 is typical what we've seen. So something that you'll see in publications that people will talk about the potential of a 2,000 liter bioreactor. So I want to just highlight how that is problematic in a moment. It's not what we're seeing, and it doesn't match some of the technical challenges that that today's manufacturers are seeing. So give me one click, Arantxa. So as I said before, if I add my DMSO, I start my cryoformulation. I now have a 2-hour window to finish the formulation, to complete the filling, and to do all the visual inspection, right? And bear in mind, if you're shipping to countries like Japan, you have to do 200% visual inspection. The second thing I want to highlight here, which I didn't mention earlier on, is that because it's a cell therapy, you're starting with a cell and you're finishing with a cell. You can't sterile filter these types of products anywhere in the manufacturing process. So they are very challenging visual inspections to complete. So the big challenge here is that if I did produce a 2,000 liter bioreactor and I had hundreds of thousands of doses, it'd be very, very difficult to visually inspect that in your 2-hour window. So what we're seeing is that the manufacturing process is staying roughly in around the 50 liter scale, because when you run the maths of how many vials will get produced at the end, that's roughly what can be inspected within 2 hours. Now it's very process dependent, but roughly the point I really want to make is that allogeneic cell therapy doesn't mean you can make limitless bioreactor scale. So this is the big challenge in these facilities, and the key thing is that visual inspection piece. It's how long is your visual inspection going to take, and that's then what is what drives your bioreactor size. Okay, Arantxa, give us the next slide. So this is the big thing, and when we know in visual inspection, there's 3 tiers of visual inspection. There's manual inspection, where you just have a board, a manual inspection station where your light is controlled, you've got dual backgrounds, but an operator is manually moving and manipulating the vial and doing the cognitive piece. Semi-automated is where you're getting a machine to help with the presentation of the vial, but you're still doing the cognitive piece yourself. Now, we haven't put up automated here yet at the moment. Again, just what we're seeing today is because of these kind of first generation allogeneic processes, with the challenge of maybe it being a difficult, cloudy visual inspection, we're not seeing many clients going straight for fully automated at the moment. And fully automated is where the presentation and the actual cognitive piece is being done by a computer system. So this is the big challenge in these facilities. How many doses have you got, and do you do it manual or do you do it with semi-automated visual inspection? And this is what's helping to size. If you can get through more visual inspection quicker, you can have a slightly bigger bioreactor. Okay, Raj, I'll take one more slide there. So, everything I've said there is quite broad. Okay, so cell therapy is a massive, massive spectrum. Okay, so you have, and I'll just give, there are easy cell therapy manufacturing processes. So these are processes where you might have a cell bank at the start. So it's all very controlled starting point. You may have a very, very simple expansion process. So you are inoculating media exactly like a traditional biopharmaceutical process, and then you're passaging up through progressively bigger bioreactors. And it's suspension cell culture. So the CAR Ts are suspension cell culture because they're blood cells. They're naturally in suspension in your body, so they don't need to be adapted. Frozen drug product. Let me explain this for a second. That obviously, are you shipping back to the clinic frozen? And if it's frozen, the problem, what that means is that it's going to have DMSO in it. And if it's got DMSO in it, if it's toxic for the cells, it might be toxic for the patient as well. Now, traditionally with the CAR Ts at the moment, is because that once they thaw that, it's being infused into your blood. So the DMSO is being diluted down, not only in your own body's volume of blood. If the cell therapy is actually being injected into a site and it's not in suspension, and the DMSO needs to get washed out before it goes into the patient. So in a kind of blood-based cell therapy, the DMSO can stay in, and then the product is a direct infusion at the clinic. A complex allogeneic cell therapies where maybe I don't have a characterized cell bank, where I do have regular donors. So I do get a donation into the clinic on a weekly basis or bi-weekly basis. And then that one donation can do many batches. The reason that's a complication is because you now need to worry about viral contamination. You now need to worry that your donors are screened, that you're doing screening of the donation at the start of your manufacturing process, to make sure that you don't bring in any unwanted viruses into the process. Complex DMSO, complex differentiation protocols. So again, some of the induced pluripotent stem cell products we're dealing with at the moment, it's not a simple expansion process. They're expanding and then they're adding a series of cytokines to differentiate the cells into whatever therapy it's going to be in the future. It might be complex because, and this is a key one, the cell therapy, it needs to behave the way it's going to behave in the human body. So again, if it's a CAR T and it's T cells, it can be suspension culture. But if it's going to be a joint in your knee, for example, if it's going to be tissue in your stem cells for a brain injury or neural cells or whatever, in those cases, it's an inherent cell in your body, so it needs to be in an adherent bioreactor system in your manufacturing process. Fresh drug product, again, as I said, if you can't wash the DMSO in the clinic, you now need to ship it at 2 to 8, which means you have a very, very challenging logistics journey, because you may only have 2 or 3 days to get it from your manufacturing process, from the completion of your manufacturing process, plus the QC release, plus the shipping to the patient. And then some of the more complex cell therapies have a bespoke medical device that the doctor needs to use to actually administer the cell therapy. So while I've given a very, very simplistic account of the 2 previous slides, it really is a broad spectrum when you get into manufacturing design. And what's really critical in any of these processes is that you need to understand the science, you need to understand what's actually happening for the patient, and then your manufacturing process needs to actually be an extension of what would naturally happen in the body. So for Arantxa's presentation and her adaptability slides coming up, we're going to just focus on it that we've a relatively simpler process. So we're going to take it as you have an autologous CAR T process, which is in suspension culture. It's going to be frozen at the very end, and all we're doing is making that an allogeneic process. Okay, over to you, Arantxa.
Okay, so we've seen some of the biggest differences between autologous and allogeneic processes and the challenges they pose. So we've also seen the trends, and clients will want us to design facilities that can host autologous processes now, but maybe allogeneic processes in the future. And as architects, we want buildings that withhold the change of time. So we are looking at designs that are flexible and modular, creating modules that are adaptable and can be configured and stacked in many ways, almost like if they were Lego bricks, to respond to site constraints and process and project requirements. So if we are to be more specific, if I take, for example, the areas that Tom has been talking about, the formulation, field, and visual inspection, I will focus in this slide in the modules that do these activities. So for clarity, I have shown in yellow the formulation and field room, and in green the visual inspection and CRF rooms. So we start with an autologous facility where we are doing the process activities in a benchtop sort of equipment. We have the formulation and fill stations in the yellow area in a biosafety cabinet. We pass through to a visual inspection, and then the product goes into the CRF or maybe into a cryo-freezer. So as we move into an allogeneic process, we start looking at automated filling isolators. We still are carrying out manual visual inspections, as Tom was explaining, and it is still at a lower scale. We pass through the manual visual inspection station. We will require more CRFs as batches are larger. So maybe at this stage, the cryo-freezers have moved to larger operations in the warehouse. And finally, as we scale up the allogeneic processes, we might look at having bigger isolators and potentially semi-automated visual inspections. And again, the CRF number will increase. So the idea is to apply this concept into every module of the facility so that the modules that take different processes can evolve with time. So we need to understand that what the requirements are now as an autologous facility, but also what they will be later as an allogeneic facility. So looking at things like the classification and the utility and technology requirements. So again, for clarity purposes, in the next couple of slides, I'm showing the process suite in dark and light blue, the formulation and filling suite in yellow, and the CRF and inspection in green. So we start with an autologous facility. We will have the blue process room here, and then the formulation and filling with benchtop scale equipment, as we said, and the biosafety cabinets. And we pass through to a manual visual inspection room then. And as we move into an allogeneic facility, then we are going to start looking at a higher demand of media, and therefore we might look at having a dedicated suite for it. So for example, maybe the dark blue becomes a solution prep dedicated to the facility. And in the process room, we can start looking at the equipment sizes changing slightly, and we start maybe seeing bigger bioreactor sizes and so on. And as I said before, the filling might happen in an automated filling isolator and passing still into a manual visual inspection and CRF room. And then we scale up again, and we can see then that we get bigger isolators, potentially bigger bioreactors, and semi-automated visual inspection. But the main thing here is to create a facility that can go from the autologous to the allogeneic processes, having as little as possible intervention in the building fabric and reducing the intervention required to make to the building elements. So having the same support areas and utility areas and creating a module that can adapt quite easily. I would also like to mention finally that there are areas in the facilities such as QC, kitchen, logistics, and warehouse that when it comes to autologous, where one patient is one batch, are quite intensive, and these can increase the headcount considerably. So if you are looking to move to an allogeneic operation, we can probably rationalize those areas. And what is key is to develop a design, again, that can transition. So the same principles of getting a modular design that can adapt over time will also apply here. So Tom, back to you.
Yeah, Arantxa, do you want to go back just two slides for a second?
Yeah.
I just use just a graphic showing the three facilities. So just one of the things that the facility on the left is a very similar to something we've designed in the last year or so, and that facility can do roughly 4,000 patients a year. Okay, so one of the things, just because I suppose we have a little bit more time on this presentation, but that facility on the left is doing 4,000 batches a year effectively. Okay, so that means if you take a traditional manufacturing facility that's only doing 50 to 100 batches a year, say, in traditional biopharmaceuticals, that facility on the left is doing 4,000. So you have to bear in mind the QC operation is huge. Okay, so that's where Andrew was highlighting there that, you know, these facilities, we've kept this presentation simple just to focus on the manufacturing spaces, but this comes with an absolutely massive QC operation that goes with it that is much bigger than traditional facilities. And because I've 4,000 batches, you know, there's a lot of logistics, a lot of material required for all those patients, and they all need to be segregated. So what's critical on an autologous facility that is doing 4,000 patients a year is that I cannot have a mix-up. You know, if I have a single mix-up, what happens there is that it's possible that two patients get their medication mixed up. So now two patients who are thinking they're getting their cancer cured or actually have received the wrong therapies, and because of graft versus host reaction, they just won't work for the two people. So one mix-up is actually two patients, and that's critical. But one of the things that a lot of our clients today are saying is that they think a lot of today's autologous first-generation cell therapy processes will actually become obsolete much sooner than traditional biopharmaceutical products. You know, so while a product might have had a 15, 20-year lifespan, they think that these processes they might get market approval, but they might be replaced within a very short period of time. So maybe they're thinking 8-9 years before an allogeneic second generation process turns up. And I think this is the key thing our clients are looking for at the moment, is that they do not want to build a manufacturing facility that is so dedicated to autologous cell therapy that as their second generation processes are coming down the line, that they're able to adapt it, that the facility will have value 20 or 30 years into the future.
I think that's amazing, Tom. Like, you know, it's not only a few years ago when we were starting doing cell therapy projects, there was a consciousness that facilities were constantly changing because the technology applied was going to be changing. But it's not only the technology, it's the fact that, you know, it will be another process in a while, or, you know, and it is critical that the facility is modular and adaptable. Yeah.
Okay, so I'll go back to that last slide then, mentioned. You know, so so what's the future going to look like? So this is just a kind of slight closing slide on my part. So, you know, so at the moment, I made a point that, you know, if you do transition to allogeneic in the future, that we probably won't see 2,000-liter bioreactors, because there's probably too many doses that you can practically fill in the short time frame that's required. So how do we get bigger bioreactors is what I'm challenging here. So, you know, do we have lower or more characterized DMSO processes where they can actually, rather than 2 hours, that they can push it to get 3 or 4 hours? So is that a low DMSO technique, or is that a non-DMSO technique? There are other cryopreservation methods out there. I'm not an expert in them, but so I think challenging how the materials used in the cryoformulation might be a benefit. So fully automated visual inspection. Again, I intentionally didn't have a graphic on it earlier on because I don't think we're seeing many of our clients using it today, but it is in general pharmaceuticals, AI and fully automated inspection is becoming more and more and more, and it no doubt will come and help with cell therapies in the future. So this, again, if we can speed up the visual inspection, then we can then fill more doses, or we can scale up our bioreactor even further. Again, just, you know, just today, you know, when you look at a modern fill finish facility for lyophilization, you know, the filling machine can do everything now. It can fill, stopper, cap, visually inspect, and then automatically, maybe not visually inspect, but can automatically load onto trays and fill into a lyophilizer, and then automatically do the lyophilization process. And at the moment, there's no equivalent for that for controlled rate freezing. There isn't a machine that will automatically take you from the filler over to the freezer, so that you can further streamline that back end again to just get very repeatable results on that DMSO through to loading your freezers. So again, that might be some technology in the future that could work. So one company we're working with is sort of looking at the following, but really it's, can you kind of, when you get your bioreactor ready for filling, that can you do a sort of scale out formulation process where, you know, if you have a, say, a 200 litre bioreactor, that 50 litres gets passed into a formulation vessel, formulated, filled, and then you keep sending aliquots forward. So the DMSO addition is happening in stages. So yes, you can get a bigger bioreactor, but you just have to be careful there that your cells from your first sublot are the same cells as your last sublot, that you don't have a quality difference between any sort of kind of partial formulation stepwise technique that's going to happen there. So that's another technique being investigated to try and help scale up these processes further. I think we're done. Arantxa, is there another slide?
Yep, no, that's it.
Okay. So I think that's us finished. So I think we're ready to take questions if.
Yes, thank you very much, Tom and Arantxa. So the first question is from Vernon A, who says, somewhere I read that in cell and gene therapy sterilization by steam or filter, both ways is not possible. Is this correct? If this is true, then how are we producing aseptic product for this particular formulation?
So it is true. So the steam sterilization bit is that because of the scale of cell therapy processes, they're exclusively single-use components. Or some companies are autoclaving some componentry, but in principle, the facility that we're dealing with now, it's everything is single-use. Even, you know, any tools for inside biosafety cabinets, etc. How they're keeping it aseptic? It's aseptic from the very get-go. So from the moment that donation arrives in the facility. So this is a very interesting point, is that when you actually look at cell therapy, if you look at traditional biopharmaceuticals, you had a drug substance process, which was the biologics piece, which was growing cells, purifying the protein. And that was all, you know, either azenic processing or bioburden control. And then that left to go to a different facility to do your aseptic formulation and filling. So the two types of facilities were separate. You had the bulk drug substance and then the filling. These cell therapies, you're growing cells from the start. So you're doing all that, plus because you can't sterile filter the cells because the cells will get caught up in the filter, that means it's aseptic from the get-go. So it's interesting that when we see professionals who are hired because they're biologics SMEs, as in the people who grow cells and purify, but then you might get other people who are aseptic SMEs from traditional fill finish type facilities, but they're now together. So the two professionals have to work together to make sure that the biologics process and all the aseptic technique is being maintained from the very moment you start the process all the way through.
Thank you. And Tom, Sammy asks, what's DMSO short for? I think you answered that.
Dimethyl sulfone, something, I can't remember.
Sulfoxide.
Dimethyl sulfoxide. Yeah. So it's a quite, so in the liquid phase, it's actually quite toxic for cells. So they have to freeze it. So in your traditional biopharma processes, you'll know that they have this out of vial condition. So your cell bank, they have to wash the media out of the cell vial as quick. So as soon as they complete the thaw, they have to wash really quickly to get rid of the DMSO because it's toxic. So here it's kind of the opposite. It's once we add it, we need to get it frozen really, really quickly. I'll just explain, you know, if you take Takeda's Allogeneic, which is the only approved allogeneic cell therapy at the moment, it is a fresh product. So they don't add DMSO. So I'll just explain this a little bit more. So when that's used to basically people with Crohn's disease, they develop holes in their colon, and what the cell does is it's used to basically plug a hole. So the cells are basically injected into a hole and it blocks the hole. But there's no blood flow. So because there's no blood flow, what it means is that if you did have a frozen product and you did just thaw it, those cells, when they're placed in the body, they're still sitting in DMSO in the body because there's no blood circulation to flush away the DMSO. So for that reason, the product has to be fresh. Now they could, okay, and this is the interesting part of cell therapies, they could say, well, why don't we freeze it? But then the hospital does a media exchange. But the problem now is that, is the doctor now a QP? Because where is it a final product? It's not a final product. It leaves the hospital, but there's another manufacturing step required in the hospital environment. So it brings up challenges to say, well, when does the pharmaceutical companies, remit end? So that's the key thing. I think there's another question coming over. The CAR T therapies, because they're being infused directly into your bloodstream, the DMSO ultimately gets diluted down by the total volume of blood in your body, so that the DMSO is below sort of toxic levels for all the cells in the patient's body. So therefore, there are some therapies that can be cryopreserved and then just be used by the clinic as is, but then there's other therapies that you have to be aware of how is it actually administered to the patient, and does the DMSO have to come out?
Thank you. So I think you answered the question from Milind, which was how the concentration of DMSO is determined. Is that right?
So yeah, I think from a medical point of view, I don't know that. So I think their standard concentration, I think it's 5%, is a fairly, and for a lot of this cell therapies, the DMSO addition has been happening for years in blood banking. It's not a new process to cryopreservation, but how they actually determine the industry norms, I don't know.
And related to that, because Decker asks, would these immunosuppressant co-therapies have side effects?
So immunosuppressants was the word I was struggling to find in the presentation. I was calling it something else. Couldn't think what the word was. Yeah, so it's interesting. Like, one of our clients has a allogeneic. It's for treating Alzheimer's disease, and it's neurons for repairing neural cells in the brain. So they're one of 3 companies doing the exact same cell therapy, and they're doing it with this immunosuppressants approach. They have a competitor who are going to do an autologous version. So that means, and that's going to be much more expensive because it's only one patient, one dose. But yes, that autologous in theory will deliver a much better outcome for the patient, because they're not now on immunosuppressants for, now, I mean, the side effects there, the obvious side effects is that if you put somebody on immunosuppressants, they're now vulnerable to other diseases, not necessarily what they're being treated for. So that is a big challenge with some allogeneic cell therapies. And then their third competitor, but the third competitor are probably 5 or 6 or 10 years away, but they're looking at doing 2 genetic modifications. So when the patients or cells arrive, they do the modification for the Alzheimer's treatment, but then they also modify that the particular target proteins that identify who the cells belong to are removed, so that the cells, when they go into the body, they don't trigger the graft versus host reaction.
Okay, and just related to that, because John Maloney, your colleague from PM Process in Dublin, asks, hey
John,
is there a reduction in treatment efficacy moving from autologous to allogeneic? And
I suppose that's the main.
Yeah, sorry. Yeah. And Kunkit says, and also to add on, is the type of cells that we can scale up in allogeneic more limited compared to autologous?
So to take John's question, I suppose that's the $1 million question. I don't think we've seen any. There are certain cell therapy companies who they're competing with their autologous rival brands, let's say, and just one company has done a better genetic modification than the other, and they're getting better efficacy with. But I don't think we've seen a company who have held the marketplace with an autologous version, and then they're now getting disrupted by an allogeneic equivalent. So I suppose there's no real market, but I mean, in principle, like, the efficacy, well, the efficacy in theory shouldn't. And what's the $1 million question is whether the immunosuppressants that they might be on, for example, cause other challenges, and as a result maybe render the treatment not as effective, be it more cost effective, but maybe not as effective in a holistic sense to the person's health. On the second question, are there certain types of cells? Yeah, so the easiest cells to transition to allogeneic are anything got to do with the bloodstream. And the reason for that is that the cells are naturally in suspension culture in your body. They're not. So if you take traditional CHO cells in biopharmaceuticals, the CHO cells in the hamster ovary is a naturally adherent cell type. So scientists have adapted that over time to make it amenable to suspension culture in bioreactors. You don't have that luxury with cell therapy because the patient wants the cell as it's going to behave in their body when it's treated. So if it's adherent in the patient, then it has to be adherent in your manufacturing process. So therefore, with just current technology today, anything that is naturally in suspension in the human body is more amenable to allogeneic scale up, and that hence would be anything to do with blood, basically.
Thank you. Now, Immaculata says, from the initial discussion, if I'm not mistaken, there are about 11% approved allogeneic products in the world. Do you know what temperature the product is stored at? Because the characteristics of stem cells are very unstable.
2 to 8. It's so that one. So 11% is 1 out of 9. Just to be clear, it's not that the numbers are still quite small. So I think there's only 9 approved cell therapies, and that's an interesting point. So I think there's a lot more approved cell therapies in the US, but the US are calling things that we might call a stem cell transplant, which, so there's a lot of procedures in the past. There were medical procedures, there were stem cell transplant or whatever, so what I'm talking about here are cells that have undergone a much more severe modification and are now more pharmaceutical than a treatment in a hospital, if that makes sense. So 11% is 1 out of 9, just to be really clear with that. It's still a very small number. And that one approved therapy is Allogeneic by Takeda, and that is shipped at 2 to 8.
Thank you.
And to clarify the question, yes, that goes through a differentiation protocol in the manufacturing process. So, during the differentiation protocol, the cell is quite unstable, as is alluded to. So again, I don't have details on how stable the final product is, but yeah, it's shipped at 2 to 8, and they were very, very elaborate. Not elaborate is the wrong word. They have a very innovative app tracking system with the hospitals. That the hospitals book the material, and then the manufacturing process triggers the starting of a batch, and then the hospital, exactly like DHL, they can track where the cell therapy is in the supply chain, mainly because the patient has to be ready in the hospital surgery, having done all their fasting and all this kind of stuff before. So that when you actually think about it, like if you think of anybody here who's gone for surgery or gone for hospital treatment, the day before you leave to get ready to go to hospital, it's still in the manufacturing facility, so it's really, really tight logistics operation.
And a follow-up question is there any formal reference or guideline for allogeneic production?
Yeah, so you have the ATMP guide to start with in Europe. Tom, the FDA has a few papers and presentations that give quite a good guidance, but there's no formal guidance from them. Tom, do you want to expand on that?
Yeah, so the ISBE are so we've recently published, which and Arantxa and I were both authors on. So they're hoping to produce a series of guides. So the first one that was produced was autologous cell therapy, and I think the next one is to be a guide on allogeneic cell therapies. So I suppose there are some guidelines from the FDA, but they're really more around clinical trials and release as opposed to manufacturing, I would say. So I suppose really because we're at the dawn of this sort of sub-industry within pharmaceuticals, I think a lot of the industry best practice guides from companies like the ISPE are really only starting, and they're going to develop from this point forward.
But just to add, Tom, I've seen later on there's a question about unidirectional flows and so on. The ATMP will talk a little bit about it, and the FDA also has a little bit, a few publications on it, although there's nothing, it's quite an open approach at the minute. Yeah, we'll get to the question when we get to it.
Thank you. And for Mahesh asks, for the media fill validation for autologous, is it possible just to qualify media fill within BSC activity, or shall we include support area like grade B, where Prodigy or CAR-T enclosed system manufacturing is going on?
So our clients media fill from the get-go. So the entire process is media fill, so that they run a sort of dummy batch with just a growth promoting media through all their closed systems so a typical autologous therapy is 10 to 12 days. So they will go through a period. And the reason for that is that there's often open manipulations at the start of the process. There can be some open manipulations in the middle of the process. And then there can be, obviously the final formulation is probably open the final filling is probably open as well. So they tend to do the media fill from the very start all the way through.
And also, can you enlighten on quality control approach for test method validations related to CGT products? Probably not. I wouldn't have a great answer, to be honest with you. I'm not an expert in QC release, particularly around any method validation. But they do tend to have complicated immunocontrol assays where they do develop cell lines that mimic the disease, so that they can then prove some level of in vitro efficacy before it leaves the facility. We have noticed some controls, and probably maybe not answering Mahesh's question on, they run dummy batches, for example. So some companies will run the actual manufacturing process, but then in parallel they'll run an unengineered, no genetic modification, so that they can do a control, so that the dummy batch acts as the control, and then they run the assay on the actual product. I probably don't have any expertise on specifically how the methods are validated. Okay, thank you. And here's one for Arantxa. Could you share layout for both type of facilities? What should be the preference while designing these facilities? Unidirectional or bidirectional?
I suppose you need to work with the client and also with the guidance available that we mentioned, but it depends if the facility as well is going to be a different process facility or just one process. And by that I mean, in an autologous facility, every cell will be a different product, but because it has a different cell as a base, but the process might be different. If you have different viral vectors in the facility, we definitely have seen where we have a multi-product or multiple viruses, and we have a unidirectional flow where from one point on, once you start getting the viral vector steps, you can't return into the entry corridor, and you have some support people in the central corridor. But it very much depends on the client and where you are geographically as well, where people are more conservative or not. Tom, I don't know if you want to add something to that.
In a very general sense, I mean, if you take biologics manufacturing, they've all moved towards ballrooms and bidirectional approaches. That's because the industry is closed in that sense, and it's low risk biologics manufacturing. In a very general sense, many companies are moving back towards, not 100% back to unidirectional, but it's generally gone further back to that direction. Some clients are trying to challenge it a little bit, but in general, I think unidirectional flow is becoming more prominent again.
But I think the key thing is that it varies client to client as well, to what extent they want that to bring the unidirectional flow. How far do you go with it? Because it's going to have a huge impact on your facility layout. Yeah. I see there's also a question there about the area classification, and it's similar. Again, you need to work with your client and see how open or closed their processes are to establish what classifications the rooms are in. We generally have seen most of our rooms have been in grade C or grade B, depends on the open process steps are in a grade B environment, maybe in a biosafety cabinet. That's for autologous. For Allogeneic, again, we have seen a grade C room with a grade B support room of it as well for open processes.
Thank you for that. And Yonhee asks, understand low or non-DMSO, would you share the trend of cryopreservation in cell therapy? A couple of years ago, I heard purified compound from leaf, ?
Yeah, again, apologies now for probably not answering the question too well. So at the moment, it's all manufacturers are using DMSO if they are preserving, and I think it's 5%. I might need to get that number correct. So that there is no, we're not seeing a trend really in facilities at the moment. They're all doing the same thing roughly. So while any any low DMSO or non-DMSO approaches, they're very much academic research at the moment. In practice, it's all DMSO, and that challenge of managing the DMSO is consistent across all manufacturers.
Okay, and a kind of medical question. Curious to know, in autologous system, can twins share the cells for treatment?
Yeah, so there's a term called homologous, which is a close relative. And again, where the line is drawn, whether somebody doesn't get graft versus host, I assume if somebody's an identical twin, then you probably shouldn't get the graft versus host reaction. But there's a categorization of some. Now it's more on the medical side rather than pharmaceutical side, but there are certain therapies that if it's a donation within the family, let's say, there's less sort of strict protocols on it. But yeah, so I think there is some, depending how close the relative is, I think that the graft versus host reaction is not as severe.
Okay, thank you. We've got 6 minutes left for anyone to unmute and ask their question or to post in the chat. This is your golden opportunity to ask our experts. And while I give you a few seconds to do that, I'd also like to put in an advert to ask everyone to please click the link that's been put into the chat, as we would very much welcome your feedback and suggestions on how we can improve future sessions. In the meantime, does anyone else have a question? Otherwise, we will let Tom continue on with his holiday and for Arantxa to get to work, I guess. Okay, so just a question about presentation slides. I think we're okay to be sharing those. And a recording of this session will be put onto the ISPE Singapore YouTube page so that people can have a look at that and review, especially if they were not able to catch the beginning of this session. So I'll just give one last chance for anyone to ask questions. Otherwise, we'll be able to let our speakers get away.
Just as a final pitch, like the ISPE had just published the Autologous Cell Therapy Facility Guide. So I think, for members of the ISPE or people who have access to those documents, I think that's it's only half of today's story, but it's worth reading. It'll give you quite an understanding of what the industry is doing today. Thank
you for that plug, Tom. So Milind asked the question if we could provide some regulations on CGT. So I think that as you've kind of answered that already.
Well, the regulations in Europe, probably the biggest document is Part 4 of Utrex, which is the HMP guide. Now, it's a very broad guide. It's covering cell therapy, gene therapy, regenerative medicine. It's covering a huge space, but that's probably the most advanced GMP available from regulators at the moment. And it's an EU Utrex regulation.
Okay, great. Any final words from Arantxa?
No, it's just thanks for the opportunity for having us, and if you have any questions, just let us know.
Yeah, everyone's very welcome to get in touch with us and to pass on the questions to you both. We'd like to thank you very much for your time this morning, and for sharing your expertise with us. And we look forward to hopefully welcoming you back one of these days in person, I hope.
Hopefully, yeah.
Yeah. So take care and stay safe, and we wish you well for the rest of the week. And to everyone that's joined us this afternoon, we'd like to thank you for your participation, and please keep us posted if you have any questions, but also we look forward to seeing you at our future events. So thank you, everybody, and have a good day, evening, whatever that might be. Take care.
Bye-bye. Thank you.
Well, thanks, guys.